Thanks a lot, Mauro, for the kind introduction, and thank you to the organizer for the opportunity of giving this speech today. Okay, today we will have a brief overview about an application of generative and artificial intelligence in data science for health, namely how to use generative models for constructing sequencing data. And first of all, let's start with an overview about what are the main issues regarding sequencing data, omics data. Okay, the first interesting issue is the fact that omics data live in a very broad high-dimensional space. And geometry in high-dimensional space is something of a kind. It's something quite different from what our intuition of spatial relationship is providing, and the geometry of this high-dimensional space is behaving very, very strangely. Here we have a few examples. Take a look at this, at the first plot on the left. Okay, there's a ball included in a square. So suppose you have a sphere of radius 1, okay, embedded of diameter 1, embedded in a cube of side 1. And let's try to express the ratio between the volumes of these elements. In dimension 1, clearly, we are comparing two segments, two segments on the real line of length 1. So the ratio is 1. If we go in dimension 2, we are comparing the square of side 1 with the circle of diameter 1. So the relation is 1 versus pi. If we go in higher dimension, the volume of the cube, of the hypercube, can be represented by 2r to the d. Okay, that's clearly, I mean, the volume of the hypercube. The volume of the hypersphere of the same diameter is expressed by the formula you can see there. If we go to the limit, you see that the limit is 0 with the increasing dimension. What does it mean? It does mean that the volume of the sphere becomes neglectable with respect to the volume of the hypercube. So there's nothing somehow virtually in the center of the cube respect to what is the total volume. So where is the total volume? The total volume goes towards the angles, okay, the vertices of the cube. This is really, really counterintuitive, if you want. And this is represented by the image on the left, on the bottom left, okay? If the black part represented the distribution of the mass of the hypercube is concentrated on the corner, while it's quite empty in the middle. Same thing happens if you consider the spherical crust, which means a sphere of diameter, of given diameter, with the included an empty sphere. So only the crust, only the outside remain. And if you compare and the limit of the volumes, the same phenomenon happens. The mass is totally concentrated on the crust. This is known as the curse of dimensionality, or the Hughes phenomenon. Okay, this is a very well-known phenomenon, and this means that somehow the distribution of the mass in high dimension is not what you're expecting. And it has a number of very strange consequences, like, I mean, random vectors have pretty much all the same length and other strange phenomena like that. In general, this has consequences also in terms of approximating function, okay? So if we don't know anything else, if we don't have simplified assumption, the sample size required to estimate a function of a number of variables to a given level of accuracy, okay, grows exponentially with a number of variables. So if we are living in a very high dimensional world, we need many, many samples to reach an adequate level of approximation of the function. And this is crucial in all machine learning application. Another important issue regarding omics data is the missing data. Missing data is a phenomenon that has always hit the problem that the omics data since they're producing, okay? Because missing values estimation is something that is known since the arrival of the microarray on the market. And in particular, missing values may occur for very different reasons. How this very seminal paper that I'm showing here, dating back to 2001 and due to the Trojanskaya group at Harvard. Okay, so for instance, insufficient resolution, image corruption. So you can always assume that any kind of sequencing data is somehow affected by missing one. And although it's a very well-known problem, this is still unsolved. If you see the other paper that I'm putting here on the screen on the right, dating just last year, February 2023, although there are recent advances due to the use of artificial intelligence, okay, missing data are still common in omics data, and they are still being produced wherever or regardless of the level of omics you are considering. Finally, the third issue that concerns omics data is the use of multi-level omics. Okay, as you well know, starting from DNA or RNA, you may construct and sequence very different levels of omics data, from methylation, proteomics, metabolite, epigenomics, and so on. So really a number of different levels. And the idea now is to reach an adequate level of integration of all these different versions of your omics level is needed to fully understand the biological process that you are studying. So these are the main three pillars that are constituting, that are affecting omics data: high dimensionality, the problem of integration, and the problem of missing data. This is perfectly coupled with the recent use of synthetic data in medicine. Okay, we know that having to deal with health data is very hard, okay, because of different issues like privacy or difficult in having the data themselves, not only omics data, but in general, even imaging or EHR data. And the idea that as provided by artificial intelligence is that generative model may help us in producing what are called synthetic data. Synthetic data are data that are very similar, in the sense that they are extracted from the very same distribution of the original data, but they do not belong to any patient. So they are free from problem of GDPR. And in general, the recent trend, and the very recent trend, the expectation is that synthetic data may have the potential to revolutionize the healthcare. However, there are a number of issues that needs to be taken into consideration if we want to be sure that the model that we are building on top of this data can be clinically relevant and clinically reliable. So in general, what are the uses of synthetic data for omics? We can use synthetic data in omics projects for model augmentation. Okay, so we can construct a number of synthetic data starting from a small number of real data to augment the training data needed to our model, for instance, deep learning model, to be more reliable and more effective. We can use synthetic data to impute, so to fill the blanks that I was mentioning before in the omics data. And we can use synthetic data to reach a good level of integration of different levels of real omics data. So differently from synthetic data, like for, in other cases, like for EHR or imaging, we have more uses for synthetic data in sequencing than for other kind of health data. Imputation is one of the, probably is the first and the oldest problem in original data, okay? In omics data, imputation or solving the problem imputation dated back to the early 2000. And the idea was to use some mathematically or statistically constructed synthetic data. What can we do these kind of things? We can do it, for instance, by averaging and the number of real data we have in the same position, like for instance, averaging the expression. Clearly, this approach is not optimal, okay, because these methods cannot take in consideration the correlation structure of the data, but at that time, 20, 25 years ago, was the best you can do. What are another problem related to this kind of issues? Is the evaluation of the goodness of these models. Evaluation of goodness of the model relate to the fact of having a good metrics, okay, for doing comparison of estimates. So this is one essential problem that started 25 years ago, but it's very fashionable even today, because obtaining a good matrix to assessing the significance of the discovery of biological results is a key for rely on good synthetic data in any kind of our application. Another alternative was the creation of virtual samples. And virtual samples, as you can see from the dates of the paper that I'm publishing, that I'm showing here, November 98, was a very old idea, okay? And the idea was to create sample that align between two real samples. Clearly, the problem was that the difference, okay, the originality of this new virtual sample was not enough to guarantee a good variation for the model themselves. So although this virtual sample may help in increasing the training set, the idea is that their variability was too limited to be effective. That's why, I mean, using virtual samples in the statistical way was a big problem since the very beginning of the creation of omics data. The idea clearly to substitute all these oldest statistical method is to use clearly the generating models coming from the AI as we know them, okay? So the idea is that we can construct a new set of synthetic data using generating models, and at the same time having four main benefits for our model: improving the performance of the training the model training, dealing with privacy issues, because clearly, as I was mentioning before, these synthetic data are very realistic, but they are not real. They do not belong to any real patient. They can enhance the dataset structure and diversity, which is the key of adding value to our model. And they can also be used for providing educational alternatives. For instance, we are developing something like that in digital pathology, constructing new imaging starting from virtual biopsies. And as you can see from the few papers I'm listing now, all this kind of synthetic data that have been introduced are stimulating the growth of a whole new branch of studying for generation model in AI for healthcare studies. What can we use? I mean, what are the main topics that we need to deal with when start in developing models, developing new algorithm for generating data? Okay. A very common trend in the last 5 years, let's say, are log linear models, okay, that when fitting to the generating data can be used to extract pattern for omics data. This is a whole thread of constructing omics data starting from real one. Moreover, generative AI can generate synthetic observation that can deal with the uncertainty of the learned representation. Moreover, we have experimented that generative-based augmentation may provide a real significant boost in classification performances. And here I'm showing a few examples, recent examples in literature that are supporting this claim that I'm telling you, okay? One first example of the new methods that are being used for constructing synthetic omics data are omics GAN. Omics GAN has been used to integrate both messenger RNA and microRNA to construct a good interaction network. This has provided a number of good applications for generating synthetic data, providing better classification performances respect to the original omics dataset. And this is a very important advance in the direction of integration of different levels of omics data. Another very interesting application is the omics to omics translation. GANs can provide a way to construct a whole synthetic new level of knowledge. So starting, for instance, from transcriptomics, we can generating data for proteomics or epigenomics or metagenomics. For instance, one of the main applications has been published in the paper you see there, where gene expression profiles has been reconstructed from DNA methylation, so epigenomic data. This has been trained on a large-scale multi-omics dataset, okay, but it was applied on a small-scale dataset, and this has been published as OmiTrans. This has provided us with not only with an integrator of data, but a real translator of omics level that is clearly a key issue for this kind of application. I mentioned before that the problem is how can I evaluate the goodness of this new synthetic data? When dealing with unsupervised application, the first thing to assess is how reliable is the distribution of the data we have constructed with respect to the original one. And the first, let's say, metric that been used are the Frechet distance, okay, that are expressing the similarity between the true and the generated data based on the last hidden layer of the perceptron, trying to discriminate tissue type, or the usual precision recall interpreted in the new omics sense, so essentially evaluating the distance between the true and the synthetic distribution, considering the dimensionality, the intrinsic dimensionality, or also F1 score, harmonic mean usually, or the adversarial accuracy. This is a very interesting application. That's one nearest neighbor accuracy, discriminating the true from generated data. And finally, the correlation score. So we can use a statistical metric like the correlation matrices of the true and generated data using Pearson. So all this kind of metric has been used to compare different distribution between the true omics data that has been used to as an input for the generating model and the obtained synthetic data. When dealing with supervised method, we also have to assess the effectiveness of the data we have provided in the models. One is the reverse validation. The reverse validation strategy is implied to compute the performance of a classifier trained only on the generated data and evaluated compared to the original data. And the other one is the data augmentation, so compute the performance of a classifier trained on true samples and only k-generated samples. Mixing up the number of true samples and the number of generated samples, you may assess in a proper way how important are the synthetic data to how meant to enhance your classifier. Together with all the metrics that are really well known, accuracy, F1 score, Braille score, one of the metrics that I'm always supporting in all supervised classification experiment is the MCC. We have proven in the year the superiority of Matthew correlation coefficient respect to other metrics for the reliable assessment of the goodness of a classifier. Why? Because Matthew correlation coefficient is one of the very few metrics that takes into consideration all the four entries of a binary confusion matrix, and its reliability relies on the fact that only when all the four entries of the confusion matrix are expressing the goodness of a model toward another, they provide a high Matthew correlation coefficient, contrary to what happens with accuracy or F1 score. Going back to the methodology, what kind of GANs we have been used for constructing and producing this kind of models? The first is the basic GAN. Okay, so we have the original omics data that are supplied to the discriminator, while an amount of white noise is provided to the generative part of the GAN that produced a synthetic kind of omics data, and the discriminator try to guess the good one versus the fake one, like a normal basic GAN. A version of it is the use of conditional GANs. Okay, use of conditional GANs where not only the noise is used as input for the generator, but also other covariates, can heavily improve the outcome of the models and providing much better synthetic data. A further improvement can come from using Wasserstein gradient penalty. Okay, so adopting instead of just a discriminator, a correlation between using a distance, a Wasserstein distance between the good and the bad example may greatly enhance the value that the synthetic data is providing. All these three GANs has been published in the literature and used for constructing synthetic data, and this last GAN variation is very helpful in this, has been proven to be probably the most efficient and the most helpful in constructing these structures. Finally, the state of the art is also using attention mechanism. Okay, so not only we are using covariates and Wasserstein distance, but an attention layer is introduced between the generator and the correlation discriminator that are responsible for injecting a priori knowledge about the structure of the data. For instance, a level of attention may take care of the fact that there are known gene interactions that can be used to construct more refined model for the synthetic data. And this gene interaction may come from correlation, so coming directly from data, or from very well-known theoretical interaction like PPA dataset. This is a typical example of what can be obtained with a general GAN. Okay, so if we start with an example with this kind of data, sorry about that. What happened? Okay, if we are only using just again on an unsupervised problem coming from public data available on TCGA, if we start from this true distribution of data, this is what we obtain using 10,000, about 10,000 sample on 20,000 gene expression levels for 33 cancer types and 24 tissue types. As you can see, the synthetic data we obtain are not really politely distributed as the previous one, as the original one. So we cannot claim that this machine has reached a good result. However, introducing an attention mechanism, the results is really amazing. Okay, the synthetic data are can be almost superposed to the original data, and this is showing the potential of these new synthetic machines in constructing very clinically reliable sequencing data of any kind. In particular, if we take a look of the metric we have mentioned before in the unsupervised case, with all these different generating methodology, we can see that the attention GAN is the one that reached the better performance and providing more diverse data and more info preserved starting from the original dataset. In the supervised case, using 8,000 synthetic example, both in a binary example, cancer or non-cancer tissue, or distinguish different kind of tissue type in a multi-class experiment, the attention GAN in both cases reaches the best result, and the results that are really enhancing the original model with respect to using only real omics data. So if we want a take-home message to remember when using synthetic omics data for the state of art of using synthetic omics data at the moment, what we can say is that using synthetic data can achieve significant gain in classification performance. Using an attention level is the best for preserving data representation. This not necessarily translate in a better performance of the model in all cases, and because the benefits of data augmentation clearly depend on the amount of the true sample. If we are starting from too small amount of original omics data, constructing synthetic data cannot reach such a great diversity on the distribution. Moreover, it has a limited impact on domain knowledge, and this is understandable since, I mean, we cannot guarantee or we cannot expect that using synthetic data may help in inducing or revealing more a priori knowledge with respect to what we have injected in the attention mechanism itself. This has been proven on very large data set, if you want, so data set with thousands of samples. What about if we restrict the use to smaller data set, which is usually the condition or the situation in which many researchers have to deal with on a daily basis? So we have tried what happens if we deal with a very restricted data set, only 200 samples, but still in high dimensional, 22,000 genes, okay? However, although the dataset is small, we still reach a good improvement in the model performance. So, although with a more limited gain, augmentation still working if we are adding synthetic data. So, although clearly the more the better in terms of the sample size, even if we are dealing with a small sample size, we can hope to achieve some good improvement. So far, I only mentioned GANs, but clearly there are more powerful models coming on the market, and stable diffusion is clearly one of the candidates. So lately, I mean recently, a couple of papers appeared where diffusion model has been used for generating gene expression profile in silico, okay? So we are coupling a training model with a denoising mechanism, and the results from the generation to the linear regressor that provide the reconstruction and construct one synthetic sample starting from the output of the generator, okay, provide a very good example of the use of these new models in construct synthetic data, which is very promising in terms of performances. If we take a look at what happens, okay, if we start from the real generator, which is in gray, okay, and we produce some synthetic data set with a stable diffusion model at different epochs, when we reach to the bottom of the epochs, we can see that the synthetic data, okay, in this multi-class problem has covered almost completely and properly the whole original distribution of the small amount of omics data we started with. So this is clearly indeed suggesting that also stable diffusion may be the new horizon for constructing synthetic data of this kind. In particular, synthetic stable diffusion model on GTX data, so public, still public available models, have a competitive performance with other mechanisms, but they provide easier, stable, and faster convergence. They are driven by covariates and domain knowledge more than GANs, okay? And they have the property of filling better the whole representation space. So essentially being more promising in terms of diversity of the generated data. Clearly, they need a larger size of the starting data set, and that can be a restraining factor for using in all situations, as we observed before. So far, I only mentioned classical, if you want, omics data, but all this method has started being used also for very recent omics level, like single cell and spatial transcriptomics. What is different in single cell and spatial transcriptomics, that not only we have the omics level, but we also have, if you want, the geographical information of the position of the cell generating the omics level. So generating synthetic data in these new domains had the additional constraint of requiring also the position, okay, of the generating cells. And this is will be probably the new frontier of using generating artificial intelligence for synthetic data in the omics domain. Clearly, although these are very recent and very, let's say, frontier techniques, they still have problems, for instance, with very old odds like imputation, okay? So blank spaces also occurs for spatial transcriptomics. In general, what happens? What is the state of the art at the moment? Most simulators at the moment are still unable to accommodate this complex design. So there's really a lot of space for people that wants to try to optimize new generator or invent new algorithm for generating synthetic data in these domains, because there will be more and more demand for this kind of features. I conclude with what we are doing in FBK with synthetic data. Not only we are start dealing with omics data, but we have a good experience in also constructing EHR synthetic data, okay? And this we have done with conditional GANs, and we have presented then at New RIPS a couple of years ago, and we are presenting, we are publishing now in a journal, probably in the next couple of weeks, okay? And also we are dealing with imaging, so digital pathology. Also here, starting with a restricted bunch of digital biopsies, we constructed a fair amount of synthetic imaging. And here too are the things to notice. Here we are using stable diffusion models for constructing them. And to comply with the evaluation problem, we use the professional pathologist to assess the reliability and the clinical meaningfulness of the image we are generating. And this is a very famous approach for everything that concerns imaging when producing synthetic data, and pathologists were somehow unable to tell the difference between synthetic and omics and real digital pathology biopsies, and this is a good indicator of the reliability of the generated data. This is why we are interested in this. We are interested in this because we are involved in a PNR project, the high HPC, high performance computing in omics, and one of the targets of this PNR project is exactly developing machine learning algorithms to analyze new omics data, including also radiomic synthetic data, in very efficient way. So coupling the synthetic production of new omics data with the construction of optimized algorithm for using in high performance computing. And this is the work we are doing together with my group, with Marco Chierici and Silvia Menchetti, that I'm acknowledged now. Okay. Finally, the last slide about what is the real challenge now for using synthetic data in medicine. I talked about omics data, but clearly I showed that there are also EHR data or imaging data. So if starting from all the clinical data of a patient, okay, we want to construct a whole virtual cohort of patients, we need to generate all the associated synthetic data, okay, clinical lab exams or radiological data or genetic or genomic data or imaging. But what's the problem? The problem is that all these data types are not independent, okay, because they are all somehow ruling the physiological or pathophysiological processes that are running in the body of the patient. So we cannot hope to virtually construct independently all this kind of data. We need a very complex method, and especially a very strong nonlinear method that can take care of all the correlation, the interaction between omics and imaging and clinical lab. So what is the problem here? This is really the challenge for the future in the use of synthetic data, if we want that this synthetic data we are producing are indeed clinically relevant. Solution, what is? The solution that is forecasted by many labs worldwide is that we need to inject a strong nonlinear causality in all generational mechanisms if we want to assess or to obtain very reliable data. And that's a challenge that we are looking forward to be involved into in the next 20 years, probably. Thanks a lot for your attention, and I hope that has been interesting. Thank you.
Thank you very much, Giuseppe, for the very, very interesting seminar.
Thank you very much. Thank you very much.
Okay.